Traditional agriculture relies on the green thumb, a blend of intuition and luck used to navigate the organic chaos of the outdoors. That reliance on nature means plants are at the mercy of the sun's broadband signal. It is a wide, unpredictable spectrum that forces a plant to guess how to allocate its energy. Inside these sterile agar vessels, we have replaced that uncertainty with calculated precision. This animated diagram illustrates narrow-band spectral control. Instead of the sun's total output, we are isolating specific wavelengths into surgical beams of light. These targeted LED arrays act as a software upgrade for plant DNA, using light as a photon scalpel to influence the plant's internal operating system. By isolating these signals, we move from observing a plant's growth to actively directing its developmental timeline. In the wild, plants have a reflexive instinct to reach upward, stretching their stems thin and fragile as they compete for the sky. We override that reflex by bathing the young sprouts in high-energy blue wavelengths. Inside the stem, light hits cryptochromes, photoreceptors that act as a biological switch. They signal the plant to stop reaching, creating immediate structural density. Zooming in, we see real-time cellular fortification, as cell walls rapidly thicken and harden into biological armor. By manipulating these blue light receptors, we manufacture a rugged, compact survivor conditioned for the stresses of the outdoors. The next phase of the hack shifts focus to the biological activity occurring beneath the surface. We influence root development by tuning the precise ratio of red to far red light. This visualizes the phytochrome equilibrium, the plant's internal shade sensor. By tipping the red to far red scale into a disproportionate state, we exploit an evolutionary loophole. This specific ratio triggers a shade avoidance panic, but with a controlled twist. Because the blue light has already locked the stem in place, the plant cannot stretch upward. Instead, it redirects every ounce of biological energy downward. Roots erupt days faster than normal, creating a dense, aggressive system optimized for immediate nutrient uptake. By weaponizing the plant's own internal panic response, we accelerate the growth cycle far beyond standard biological timelines. Once the growth cycle is complete, automated trays move the plantlets out of the spectral chambers and toward the facility's airlock. The clinical violet and crimson lights fade to neutral white. The programming phase is over. These plants possess a structural density and root architecture absent in their wild counterparts. They have been physically restructured for durability. Because we manipulated the light today, these plants are already prepared to withstand the heat, the wind, and the drought of tomorrow. We call this cellular engineering, a data-driven replacement for the traditional green thumb. We no longer hope for survival, we engineer it. This is the mastery of the invisible architecture of growth.